Hi, you guys. Thank you so much for joining us for our first ever Eye on Research mini seminar. So if you've been part of our hybrid I Believe seminars in the fall, this is kind of just something we wanted to bring to you guys after the fact of these different cancer research seminars and conventions that happen all across the globe often. And so we've got doctors that some of you guys are going to be familiar with, Dr. Katz, Dr. Stephen Katz, Dr. Hamid, and Dr. Richard Carvajal, who are joining us from their various areas to talk to you guys about what they heard at ASCO and AACR, which are then the two main cancer conventions that have been happening this last year. So thank you guys for joining us. If you guys have questions, make sure to drop them in the chat. Keep in mind, we can't answer any personal medical questions, but we can answer questions the best we can when we have Q&A the last 10 minutes of this session. So thank you guys for your time. I'm going to introduce our speakers and we'll get started. First up, we're going to have Dr. Katz, who is the Chief Medical Officer from TriSalus Life Sciences. He's going to be presenting a few slides and some data on the PERIO-1 trial. And then we're going to have Dr. Hamid and Dr. Richard Carvajal. Dr. Hamid is from the Angeles Clinic and Research Institute, and Dr. Carvajal is from the Northwell Health Cancer Institute. So take down those titles if you guys want to get in touch with them. If you need a good medical oncologist, these are good guys. Okay, well, Dr. Katz, I'm going to turn it over to you if that's okay.
Yeah, thanks so much. And thanks for the opportunity to speak today. Trying to make sure the slides are coming through okay. Great. So I'm going to talk to you about a clinical trial that my company, TriSalus Life Sciences, is running. It's called the PERIO-1 study, in which we are investigating an immunotherapy drug called SD101, which is something called the toll-like receptor 9 agonist, which is a drug that's capable of stimulating the immune system very broadly. And it does some other things which we think are very helpful within the liver specifically. Obviously, for patients with stage 4 uveal melanoma, the liver is often where the problems arise. And so we chose to focus specifically on uveal melanoma with liver metastases. And we're also using a very innovative and we think interesting delivery technology, a catheter system used by interventional radiologists, which we think can improve the ability of drugs like SD101 to get into liver metastases, which again affect uveal melanoma patients far too often. So I'm just going to begin by covering the rationale for focusing the immunotherapy approach on the liver, which we think is critical to this program's success. And so the liver presents, or liver tumors present, two very important problems, one of which we think is underappreciated, which is high intratumoral pressure, which limits drug delivery. Some data indicate that when you give a drug intravenously, which is typically what we do with oncology patients, less than 1% of the drug may actually get into the tumor in meaningful concentrations. And the second problem is that the liver as a whole is a very immunosuppressive organ. It's programmed to shut the immune system down. And when we have liver metastases within this organ, the situation is exacerbated or it gets worse. So it makes it very difficult for patients with liver tumors and liver metastases from uveal melanoma to respond to immunotherapy drugs like checkpoint inhibitors. So what we're doing in our clinical trial is we're giving the drug SD101 into the liver in a very focused way, using the arteries that feed the tumors to try to reverse this immunosuppression to give patients a better chance of responding to drugs like checkpoint inhibitors. Now, the immune system in the liver, as I mentioned, is shut down, and this is a very broad problem. Many cell types contribute to this. But we think one cell type in particular, called MDSC, or myeloid-derived suppressor cells, are key orchestrators of checkpoint inhibitor failure in uveal melanoma patients, and we'll talk about that more in a bit. Now, one thing we've learned about patients who are getting checkpoint inhibitors, and this is data from several different diseases, including cutaneous melanoma, lung cancer, and so on, patients who have liver metastases are much less likely to respond to checkpoint inhibitors relative to patients who may have disease in other organs, but not the liver. So we believe that there's something about having tumors in the liver that drives immunotherapy failure, drives immune suppression, not only within the liver, but potentially throughout the patient's body. So targeting liver metastases, we think, is a very rational strategy for driving better responsiveness to checkpoint inhibitors based on this data. And so now let's move on to the delivery system that we're using in the PERIO-1 study. So as I mentioned, we're giving the drug SD101 in a very focused way into the arteries that are feeding the liver metastases. We think this has a couple of advantages. One, we think it enables the physicians to get higher concentrations of the drug into the liver tumors and the liver as a whole, where we think it needs to be. And it also minimizes off-target exposure. So you're not stimulating the immune system in places where you may not want that to happen, which may be helpful from a safety perspective. Now, the device, as I mentioned, is used by interventional radiologists. If you look at this larger image toward the center of the slide, you can see that the device or catheter is typically inserted into an artery in the groin called the femoral artery. It can also go into an artery in the arm, and then the physician can place the catheter into the arteries feeding the liver and the liver tumors. What's different about this device, if you look to the left, it has what we call a smart valve, which is a very dynamic structure that's opening and closing in sync with the patient's heart. So with every heartbeat, it collapses and then it expands. And so this cyclical activity creates more optimal pressure within the blood vessel, and it redirects or diverts blood flow to the tumors relative to the normal liver. So we believe this optimizes delivery of the drug into the liver metastases. And we've actually studied this compared to standard catheters in a model called an oncopig, which is a pig within which we can grow liver tumors. And we found that we can improve the delivery of this drug, SD101, more than twofold when we use that specialized device that I showed you with the smart valve relative to what can be achieved with a standard catheter. So this is part of the rationale for doing the PERIO-1 study and the way that we're conducting it. Now let's talk a little bit about the drug. So I mentioned that immunosuppression in the liver and within liver metastases is a significant barrier to patients with uveal melanoma in terms of responding to checkpoint inhibitors. And so this is a very complicated problem. And what I'm showing you here is that many cell types are involved in causing this immunosuppression. And so we want to activate many different cell types. But I mentioned that there was one cell type that may be really important in creating this propensity of failure to checkpoint inhibitors, and that's the MDSC or myeloid-derived suppressor cells. So one of the reasons we chose to study SD101 in uveal melanoma patients is we believe it has a dual mechanism of action. One, it has the ability to stimulate the immune system broadly. Many cell types are activated by the drug. Many cytokines are stimulated by the drug, which we think addresses the complexity of the issue. Two, we've published and demonstrated in patients that this drug can bind to its receptor, TLR9, on MDSC, and it can eliminate those cells by causing what we call apoptosis or cell death. So you get the broad immune stimulation, and you can eliminate the myeloid-derived suppressor cells. So that's why we think it's potentially an ideal drug for combining with checkpoint inhibitors for patients with uveal melanoma. And so now I'm going to cover a little bit more about the trial, and this is basically a recap of data that was presented by Dr. Montazeri from MGH at the ASCO meeting in June. Here are the clinical sites that are currently participating in our trials. We actually have 3 studies open and enrolling. They're called the PERIO studies, which is for pressure-enabled regional immuno-oncology, capturing how that device that I showed you is working. And so uveal melanoma is the PERIO-1 study. It's the first study that we started. We also have another study called PERIO-2 for hepatocellular cancer and cholangiocarcinoma, and PERIO-3 for pancreas cancer. But we're going to focus on PERIO-1 today. We've enrolled and treated over 70 patients with over 270 SD101 infusions to date across these studies. And I'm also showing you the countries that we're going to be moving into later this year with the PERIO-1 program. And so an overview of the study design. So patients get 6 doses of SD101, and these doses are being given by interventional radiologists in the interventional radiology suite. It's an outpatient procedure, and the patients are getting 3 doses a week apart. Then there's about a month off, and then they come back for another 3 doses, so 6 total doses. And during this time, they're getting a checkpoint inhibitor, Opdivo or Keytruda, or the combination of Ipi and Nivo during this period. And those drugs are given intravenously as they typically would be. And overall, these procedures have been very well tolerated by the patients. As I'll show you, the safety data is very reassuring. And just the logistics of it have also been well tolerated by the patients and very well managed by the sites. We started with what we call cohort A, which was a single agent dose escalation experience, which means SD101 alone, no checkpoint inhibitor, just to see if giving that type of drug into the liver directly, which had never been done before, would be safe and well tolerated. And it was. And then we moved into cohort B, which is when we brought in Opdivo or nivolumab in combination with the SD101. And we've also opened what we call cohort C, which is SD101 with the dual checkpoint inhibitor regimen, nivolumab and ipilimumab, or Opdivo and Yervoy. Now, this slide just highlights the types of patients that we're enrolling. So again, these are all stage 4 uveal melanoma patients with liver metastases. And what this bar graph is showing you is that we're enrolling, for the most part, heavily pretreated patients, so that you can see here, most of the patients are second line and beyond, including patients third, fourth, and sixth line. So just giving you a sense of the treatment experience of the patients that were included in the PERIO-1 study to date. Here's the safety table that was presented at the ASCO meeting, and I'm not going to go through all of this detail. I'm just going to point out that some of the common events, which were generally minor, are what you would expect with an immunotherapy drug, fevers, fatigue, etc. But most of these were minor and easily resolved, either with no treatment or very minimal intervention. I think the main point I want to make is that the serious adverse event rate related to the drugs, either SD101 or checkpoint, was only 5%. So this is very well tolerated. And we think the delivery approach is part of the reason why. Again, concentrating the drug in the liver, minimizing the exposure elsewhere in the body, we think creates a favorable safety profile. And as I'll show you on subsequent slides, this is coming along with very promising immunotherapy or immune activity. Now, this is the pharmacokinetic data, which in plain English is just measuring the levels of the drug on the left in the blood or the serum, on the right in the liver tissue based on biopsies. And so looking at the left, which is drug levels in the blood or the serum, what we found is that even at our highest dose that we escalated to, which is 8 milligrams, in each of these peaks or collection of peaks, which is one of the 6 doses, and multiple peaks representing multiple patients at each time point, the drug is essentially gone after 2 hours in more than 95% of patients, meaning we're minimizing the exposure outside of the liver, which was the goal. And again, why we think we're seeing a good safety profile thus far. And on the right, I'm showing you that we're getting very high drug levels in the liver when we do biopsies pre and post infusion. So in short, we're achieving the goal that we set out to achieve in using this delivery approach, minimizing systemic exposure and maximizing exposure within the liver, which has all been well tolerated thus far. And here's an example of the immune effects that we're seeing in patients with uveal melanoma. So on the top, what I'm showing you is immunofluorescence. And so we're staining a liver metastasis biopsy before SD101 and after SD101 for T cells. And so the T cells are being stained with green and red. And this is important because if a checkpoint inhibitor is going to work, we think that you need to have a high density or a high number of activated or exhausted T cells in the tumor. After all, checkpoint inhibitors are targeting T cells for the most part and not tumor cells, unlike conventional cancer drugs. And so what we see on these images is that before SD101 was given to this patient, very few T cells were present in the liver metastasis. And when we gave the SD101, and this was approximately 57 days after the first 3 doses, we're seeing a significant or a marked increase in the presence of cytotoxic T cells in the tumor. Again, the green and the red staining, indicating that the drug is having this type of broad immune effect, inducing T cells to come in, which is what we hope to see. And on the bottom, I'm showing you a bar graph, which is summarizing data from 5 patients liver metastasis biopsies where we measured the number of these myeloid-derived suppressor cells. And in all 5 patients, we saw a decrease. And in 2 of the patients, complete clearance of the myeloid-derived suppressor cells after getting SD101. So this is some evidence of this dual mechanism of action that I alluded to earlier. And if you want to get more information about the immune effects of drugs like SD101, you can look at how gene expression is changing in immune cells. And we use something called nanostring, which measures how individual genes are being increased or decreased in response to a therapy, in this case, SD101. And when we looked at biopsy samples from liver metastases, we first focused on genes that typically are associated with MDSC immunosuppressive function. And we found that after treatment with SD101, these genes went down, which goes along with those cells being eliminated, which is what I showed you on the previous slide. And then we also looked at genes that are typically associated with T cells being active and participating in a productive immune response. And we saw that several of those genes were increasing in patients after treatment with SD101. So again, this dual mechanism is playing out. The next thing we wanted to figure out, okay, so we're getting a good immune effect in the liver, it's being well tolerated, but sometimes patients have tumors outside the liver as well. And so we actually looked at these gene expression changes in immune cells outside of the liver. We collected immune cells from the blood and we did the same test, nanostring, and we also did something called flow cytometry. And here we're actually showing you flow cytometry data, which is measuring protein levels on individual immune cells. And we found that in the blood, after SD101 was given, a cell type called natural killer cells expanded, which can help with immune responses. And there were certain signs related to immunosuppression that actually went down. And we also have seen increases in cytokines, which are proteins produced by immune cells, go up in the blood or the serum, even though the drug is really being concentrated in the liver, which we think indicates that there may be a very strong immune response in the liver such that it's spilling over into the blood systemically in a favorable way. Now, one of the things that we focus on in uveal melanoma patients is called circulating tumor DNA or ctDNA, which is a measure of tumor burden. And with other drugs like Kimmtrak from Immunocore, this test has been correlated or linked to favorable outcomes of long-term survival. So when we see circulating tumor DNA go down, we believe that's an indicator that the patient's more likely to have a favorable outcome and a longer survival time. And what we're seeing in the PERIO-1 study is that the majority of patients treated with SD101 and checkpoint inhibitor are seeing drops in the circulating tumor DNA after treatment, which we think is a favorable sign that the treatment's having a positive impact on the disease in these patients. But ultimately, it comes down to survival and disease control and how the patients are doing clinically. So this is a swim plot in which we're looking at individual patients. Each of these yellow bars is a patient, and we're measuring survival time on the x-axis in days. And we're looking at the different dose levels and cohorts. So I'm going to call your attention to the green group, which is cohort B at 2 milligrams. These are the patients who got SD101 with Nivo at the lowest dose level. And what you can see quickly by looking at this graph is most of these patients are tracking out beyond a year at this point. So in this heavily pretreated group of patients, this is a favorable sign. And we're starting to see responses now, including a partial response and multiple minor responses. One of the things that we've learned about this drug and immunotherapy in general is that tumor shrinkage can actually take much longer than you would expect relative to chemotherapy or small molecule inhibitors. The tumors might actually swell initially from the immune response and inflammation. So it can take more time for shrinkage and classic responses to evolve. And we've actually seen that with this drug, where responses can actually take more than a year to occur after prolonged stability or disease control. So we're starting to see a favorable clinical profile emerge. And these patients at the lower dose level are just earlier in follow-up. And we're hoping that these bars will track out even further as these patients continue on the study. And so to summarize what I've told you, we feel that the data from the PERIO-1 study to date are supportive of the SD101 dual mechanism of action, the broad immune effect, and the MDSC elimination. Thus far, SD101 given directly into the liver with what we call pressure-enabled drug delivery, that specialized device with the smart valve that I showed you. It's all been well tolerated with desired pharmacokinetic or concentration effects with a serious adverse event rate related to the drug of 5%. The circulating tumor DNA and that swim plot showing the survival times we think is encouraging for what we're going to see at the higher doses and when we move into phase 2 toward the later part of this year. Thank you for your attention.
Thank you so much, Dr. Katz. I feel like this was just some incredible data and really just very promising. So thank you. And thank you for the way that you explain things. I know we had a couple of questions. So if you're able to take one question, do you have time for that?
Absolutely.
Actually, that question is no longer relevant. Never mind. My main question that I had was just, can you define the T cells? Like, what are T cells? And like, because they can be in a healthy cell and in a cancer cell, correct?
Yeah, so T cells are a part of our immune system. You know, normally they help us fight viruses and other things that can cause us harm, but our bodies also try to mount immune responses to tumors. So typically T cells are circulating in the blood, they're in lymph nodes, in the spleen, there are also a lot of them in the liver. And the problem is that cancer has ways of shutting T cell function down, and that's the immunosuppression problem I mentioned earlier. That MDSC cell type, they are very good at turning T cells off. Checkpoint inhibitors are designed to turn T cells back on, which is why those drugs work so beautifully in so many different indications. But within the liver, we think the immunosuppressive problem is perhaps a bit more profound than in other body sites. And so that the checkpoint inhibitors need a little more help to have their effect, to get those T cells into the tumor where they can work. And that's where we hope SD101 will come in.
Okay, awesome. Well, thank you so much for your time.
Thank you.
We're going to go ahead and excuse Dr. Katz and let him get on with his weekend. So have a good rest of your weekend and good rest of the summer.
Thank you so
Dr.
much.
Hamid and Dr. Carvajal are going to chat a little bit about Replimune, which is a phase 1 trial, I believe, in the States. Dr. Hamid has some slides as well, so I'm going to let them go ahead.
Can you hear me?
There we go.
All right. So I want to thank you for the opportunity to not only come and talk about what is going on with ocular melanoma these days, as we're making great strides, but the ability to just sit with a bunch of colleagues and friends who I have a huge amount of respect for, specifically Dr. Carvajal. Richard and I have been doing this for a long time, as you can see by the grays, but I think it's important to note that when we first met, we were just trying to figure out what to do with melanoma itself, and all of those breakthroughs are leading towards greater success in ocular melanoma. We'll start a little bit of talking about the Replimune drugs, but I think it's important to note that these are relatable to any type of immune therapies. You just have to find the right way to get them to melanoma and the right way to get response. These are my contacts. Please feel free to contact me directly if you need all that. And I would say that is help with ocular melanoma, Angeles Clinic there. So a necessary plug for him, but you can reach out to him directly also on the West Coast. We've made huge strides in approval of a drug for metastatic melanoma and tebentafusp, but we're now looking at how to get directly into the tumors and how to increase the efforts that we've had. Replimune drugs are tumor-directed oncolytic immunotherapy, and this is how they work. They are viruses. This is a herpes simplex virus that's modified to express a protein and immune-stimulating proteins. So these are injected directly to the tumors. Some of you will remember that this is how T-VEC or telemagene lehepravec was made, but the payloads are different in these drugs. And what they do is when injected, they will directly go into only the tumor tissue, and they will spare normal tissue. And then this host antiviral response will allow virus replication. Those viruses, I always say, are like rock stars that go into a hotel room. They break everything apart and they destroy the cells. The other things they do in those cells is they replicate, so there are more of these viruses. So the progeny is released. They can cause local inflammation. They can then traffic into other cells, but also this local inflammation can cause T cells to come in and cause a strong, durable, systemic anti-tumor immune response. So it can work directly in the injected tumor and also into distant tumors. One of the areas where we've tried and wanted to really be successful is injecting directly into the liver. As you've heard from Dr. Katz, the problems have been that it's hard to get to the liver. Those liver tumors are harder to get an immune response. And as we have published and presented, pound for pound, stage for stage, if you have the same amount of tumor and in a patient, if those patients have liver metastases, their outcomes are worse. So working together to destroy the liver metastases and to create an immune response is important. Going to and coming up, this was the data presented, just to say that you can see here there's a lot of effort has been gone into this drug and similar drugs. This is not supposed to be a rep for Replimune. It is for the whole idea of oncolytic or directly injected therapeutics to be in ocular melanoma, whether it's injected with a catheter, like Dr. Katz said, injected with a needle, whether it's an oncolytic virus or other. But the way that these drugs, this oncolytic therapy has been modified is initially RP1 is just the virus itself with these modifications. Most importantly, the insertion of a protein that improves direct tumor killing and increases immunogenic cell death, which is just a lot to say, that improves the way that these viruses destroy the cells and bring in T cells. And as we move up, there are payloads that can be put into these viruses that when they are replicating, these payloads can then be expressed. So important to get in RP2 that the addition is just an anti-CTLA-4 antibody. And a lot of you are familiar with that. Anti-CTLA-4 antibodies are like ipilimumab, which is Yervoy, or tremelimumab, and they have shown benefit even in ocular melanoma. So why would we want to put it directly into a virus or get it directly into an area? One, you can ensure that the drug is where the T cells are and where the tumor is. And number two, one that's not been focused on a lot, how can we get rid of these viruses? Well, you can take antivirus and medications. So let's say you give this drug and a patient has a side effect of an anti-CTLA-4 antibody. By giving an antiviral, you can cause those cells to die and control the toxicity. And there is upcoming data with RP3, which adds also CD40 ligand, 4-1BB ligand. These are other checkpoint inhibitors. These are other drugs that help to either stimulate or remove the inhibition of the T cell by the tumor. There was lots of data presented here, but I'll focus on the fact that in a handful of patients, there was a response, a 30% response rate, and these were all patients who had failed anti-PD-1 therapy. So when you think about how we're treating ocular melanoma these days, it's usually with a combination of nivolumab, ipilimumab, or a nivolumab or pembrolizumab and another drug. This is in patients who have failed that or their disease has grown. Now we're showing that there are drugs that can help restore that immune sensitivity and response. And these patients were either treated with monotherapy or a combination with anti-PD-1, which could have been, which was nivolumab, sorry. Oh, It's always important to talk to physicians and to patients and colleagues with pictures, okay? The pictures really are indicative of the benefit we have. And there's a lot to see here. You can see that this is in perspective. So in screening 3 months, 6 months, the red circles are areas where there was tumor and it was directly injected into the tumor. So we are, unlike some of the initial experiences with oncolytics where in skin melanoma, we injected under the arm, in a lymph node, or in the skin, this has shown the ability not only to be injected directly into the liver, but to be injected directly into the liver multiple times and to show response in a directly injected tumor, but also in other areas that were not injected. So this is how those immune system to go distant areas, whether it be in the liver, lung, or so. I think that's the power in this one. The same here in this patient who was injected directly into a tumor into the left eye. As you can see, the CT scan shows after injection shrinkage, and the picture of the above the orbit of the eye shows a response, but not only response in the local area. Where some of these direct injectable therapies have failed is that they have failed to go into a distant area. And as you can see on the right side, that this therapy directly injected into the eye area has shown that it traveled and caused response in a patient who had bony metastases, so tumor in the bone, and has gotten what we wanted, a complete response on PET scan. To be noted also, this patient saw prior Opdivo. Again, you can see the same here. So just want to show, and this is a patient who got not only RP2 and Opdivo, but also Opdivo or nivolumab. What about safety? Well, I don't want to get onto something that can cause significant immune toxicity, hospitalizations, etc. What we saw here, really low grade, which means not very morbid and very manageable toxicities are of low blood pressure, chills, fever, rash, etc. And there was only one significant side effect, which was low blood pressure, and nothing that was hospitalizationable. So, these drugs and this drug itself show good tolerability in patients with liver metastases, and also the ability to be re-injected multiple times to improve the response. And that's where this is. Now, Dr. Carvajal will tell you, when I talk about melanoma, I always tell you that melanoma is the standard for which we educate our friends and colleagues in other solid tumors. So various types of tumors have responded with this drug with direct injection. So hopefully the breakthroughs we make now in ocular melanoma can help our patients with colorectal cancer, with gallbladder cancer, any other cancer that can go to the liver, and immunotherapy is valuable. So in conclusion, I've shown you not just the promising for our patients received what are standards, which are very tolerable, and it works in what we call really a cold tumors that are not immunogenic themselves, like ocular melanoma. And I look forward to a bigger study, which is upcoming in ocular melanoma, and also the data with RP3, adding more payloads into this therapy. So with that, what I would like to do is now go back and bring in Dr. Carvajal and have a good discussion about, your thoughts, and we're in this phase, which are early drug developmental.
Yeah, Omid, I mean, the data that you and Dr. Katz have presented is so phenomenal. Can you guys hear me okay?
Yeah.
Audio's okay? Yeah, because again, yeah, Dr. Hamid and I go back a long ways and have been doing these trials for a long time. And he probably remembers, like I do, when we used to put people on these phase 1 clinical trials, which is, these are trials where we're testing things for the first time in people, or testing these new combination of treatments for the first time in people. And even though we offered those with therapeutic intent, right? We said, look, Mr. Mrs. So-and-so, we think this is something that might help, but we're not sure. In reality, back in 2000, 2005, even, the likelihood of really helping our patients was under 5%. And now you're seeing this data from Dr. Katz, right? First time we're injecting this SD101 drug into the liver and combining that with checkpoint blockade, or the data from Dr. Hamid, the Replimune data, where we're injecting tumor with this virus, and we're seeing these amazing responses, a response rate, the likelihood of major shrinkage of 30% in this disease, where we used to be very happy with a response rate of 5 or 10%, it's remarkable. And so, for the patients, the advocates, the people who are listening, I think we're not doing good enough. We have a lot to do, but God, there's some really nice progress being made.
What I find extremely positive here is it's not just that we have 1 or 2 drugs. We have a multitude of trials and drugs. So when we were starting out in phase 1, we would go look at for something that would just have a modicum of hope in uveal melanoma. Now, you've listened to 2 drugs, but Dr. Moser and Dr. McKean are going to give you 12 more drugs. And I could think off the top of my head, where drug development has come in ocular melanoma. Number 1, we have drugs that have shown benefit. If you look at tebentafusp, it's shown benefit. But not only have we haven't stopped there. We now have a next generation drug that's similar to that, which targets PRAME, which is the same type of immune antibody, targets another thing on ocular melanoma, and it's showing activity. And we're taking those drugs and we're putting them together. So these drugs that you're seeing here can be combined, like we combine them with nivolumab. And also the paradigms that are coming out in skin melanoma, what we call cutaneous melanoma, are now being utilized in uveal melanoma also, which is neoadjuvant. Well, what can we do before we take that ocular melanoma out to stimulate immune response? Adjuvant. What can we do to decrease the risks of recurrence? And all of these drugs will step in and help.
Yeah. So I would caution. So I do think that we're definitely in a new era of, promise for this disease. One thing that I always like to talk about is, how are we defining benefit with these, right? So the things that, Dr. Hamid was talking about is response rate. We're seeing these great response rates. We're shrinking tumor in uveal melanoma, which is fantastic, right? And with Dr. Katz, he was showing those swimmer's plots, people really doing well for a long, long period of time. And both of those are fantastic. But all of those are what we call kind of surrogates, I guess, for what we ultimately want to do, which is help people live longer and ultimately cure disease, right? And so what we're seeing are really promising signals of activity, but we definitely have more to do, right? We can't just look at this Replimune data and say, wow, this is it. I think this is really promising, but we definitely have to investigate it further. And we need the help of everyone on the call. We need the help of the patients, the family members, and advocates, because we need people to go on these promising trials, really to figure out, is this really as good as we think it is?
Yeah, I would agree with you. And I would say that in ocular melanoma, despite these breakthroughs, we are still at a place where for the majority of patients, the optimal first-line therapy should be consideration for clinical trial. And definitely second line and further, you should be utilizing the physicians who have expertise as your caregivers, your collaborators with your local oncologists, in order to find these trials and to benefit.
Mm-hmm. And I think it's on us to make sure that there's access for these trials throughout. A lot of people are traveling for these trials, which is amazing. I mean, the things that our patients put up with the travel they do, all these biopsies and procedures they put up with. It's really amazing. But God, I would love to make it easier for them. I'd love, broader access. And that's on us, and we are thinking about that.
Agreed. It's not just it's not just hard to get to find these trials. It's hard to get these trials up and running. If you look at the data and what Dr. Katz has presented, and some of the data that comes from Delcath and our colleagues who are doing the immunoembolizations, these are trials that require multiple different physicians with specialties, including interventional radiologists. They require significant budgeting. They require significant hospital resource. As a researcher, that makes me feel uncomfortable because it's a lot of work. But as for a patient, it should make you feel supported in a major way, and would make you want to look for these trials and these options, because I just find it so much deja vu in what we were saying to our patients with skin melanomas, that find the trials. Number one, find the trials. Number two, we're making huge breakthroughs. Number three, these breakthroughs are happening in rapid fashion at the speed of light. And number four, we are changing your survival through these phase 1 trials, period.
Yeah, it's really great. Now, I think, what was really important, I know Dr. Katz went through all of these, a lot of graphs and a lot of figures, which are, and he described it very, very well. It's complicated. But I guess the point is, when we ask you to do these things, when we ask you to do these multiple biopsies and spend all day in our infusion chair and draw these blood tests, it's really to figure out, are things working? Is the treatment doing what we expect it to do, right? Is it working? If it's not working, why is it not working? And that's sort of the information that we're trying to get from these trials, right? We spend a lot of time trying to figure out how to design these trials smartly, right? So we learn as much as possible with the fewest number of people we have to put on the trials, right? So we can really rapidly figure out which ones are promising enough to move forward, which ones maybe should we just say, no, this is not going to be it. Or if we have to pivot, right? If this kind of works, but we have to add another drug to it. And that's kind of the thought process as we're designing and conducting these trials that we go through.
Right. And for me, I would say, and I think, Rich, you can remember that there are some patients who have these phenomenal responses and benefits, and we now have the tools to understand why. We have the circulating tumor DNA, we have the next generation sequencing of those patients, we have the predictive markers in the tumor and the tumor microenvironment, and ultimately why something works in this person we can use to then have a specific cassettes for each patient. Well, you're so much like this patient, you need to get this. You're so much like this patient, you need to get that. So all the people that work behind the scenes and for these predictive markers and these correlates of response are important. And we shouldn't, leave this meeting without saying that the support and that comes through philanthropy, through patients, and through places like a Cure in Sight are so important to the work that we do collaboratively.
Yeah, and I have to thank a Cure in Sight for putting this together. I mean, something I say frequently and often is that this is it's a team effort, right? It's not just the doctors and the drug companies trying to put these things together, but this is a partnership with everyone on this call, with the laboratory scientists, with advocacy.
No, thank you. We can't thank you guys enough for being here, for sure. I do have just a couple of questions, just maybe to try and clarify, and it looks like our Q&A is starting to come in. This patient is just saying, how, as a doctor, like how do you help your patients decide between so many promising trials? Obviously, the science can be complicated, like you said, for the average person, and that can be overwhelming for the patient to look at some of this data and go, well, you know, my doctor or some other doctor is telling me I should do IDE, 96, or I don't know, I think I'm messing up that number, but when you have conflicting opinions, how do you kind of suggest that a patient or a patient doctor combo go into that decision-making process?
Do you want to go first?
Sure. I think it's partly you can look at the patient, the extent of involvement with their ocular melanoma, how they are, what we call their performance status, their ability to go to the places where these trials are. And I would also say, that it's not just a one-off. If the first thing doesn't work, the second thing may. Our patients with ocular melanoma now are utilizing multiple therapies prospectively.
Yeah, that's very important.
Yeah, it's a really great question, right? How do you look at this entire scope of options and prioritize them? That's really the question. And I think, in my mind, what you want to do is, if you could, figure out, what is most likely to help out of that entire list. If you could figure that out, what's most likely to help, and then can you get access to that? For the patients, typically, the ones that are most likely to help are the one where there's the most data. Where have we treated the most number of uveal melanoma patients and seen the best outcomes, right? And those treatments tend to be in more advanced clinical trials, right? So simplistically, the phase 3 trials tend to have more data supporting them. And so you can think if there's a good phase 3 clinical trial for uveal melanoma, there's probably really good data to support that, followed by a phase 2, followed by a phase 1. It's not always the case, but I think that's a nice generality. But the feasibility is also really important, right? Because if the best trial is in Greenland, can you go to Greenland?
Yeah, such a good point. This other question or other part of the question was, if you're presented with multiple options as a first-line treatment, there's certain trials we know are first-line only, and then we have things like the Perio one where they say they will take any line of treatment, first line or all the way to the sixth line of treatment. So how do you guys as doctors make decisions with those kinds of situations? Because obviously a first line treatment can be important if you want to have that opportunity and the data is there, but sometimes I think time is not always on the side of a first line treatment.
Well,
yeah, so this is where it's really important. Oh, I was going to say, this is where it's really important to see someone like Dr. Hamid or Dr. McKean or Dr. Moser, someone who actually knows the entire landscape, right? Because most oncologists will not know, which treatments are going to exclude for subsequent ones, right? It's going to be hard for the patient to figure that out, him or herself.
Yeah, I would agree with you. I think, being from LA, I would say that ocular melanoma is a rare tumor. It's a boutique tumor. You have to go to someone who focuses on it, because our colleagues know more about breast, lung, colorectal, prostate cancer, because those are the more common ones, than we do. But we know about this one little cancer that has a low incidence, but to you is important. And ultimately, we know that there may be better options than what is an FDA-approved option, whether it's that FDA-approved option plus something else in a clinical trial. And the majority of us who consult are looking not only to help you find the trial, but to help you succeed locally. So if that benefit is to go back to your local oncologist and get therapy, we can help make decisions at certain key forks in the road. What's the right first thing? Is it available near you or somewhere else? What else can we add if we need to? How can we get through toxicities? Because some of these trials and these drugs have novel toxicities that have not made it into the community, and what to do if you're looking for the next thing. So I've had a lot of patients who come and go, come back and forth. And I would say that one of the best things, if not the only thing that's come out from this post-pandemic lifestyle, is the ability to go see Dr. Carvajal or Dr. McKean or Dr. Moser in this fashion, through the phone, through a Zoom, where it does not take, historically it was, I'm going to get on a plane, I'm going to go to New York, and I'm going to have to come back. And that's a lot financially and time-wise. But now it is almost the easiest to get an expert opinion. So one of the things that I tell patients is find an expert who will be part of your team, along with your family, your spouse, and your local oncologist.
I feel like that's so important. This one, Kathy is asking Dr. McKean, who and where are they? Dr. McKean is coming on next, so we will make sure you know where she is from, but she's in Tennessee. So Dr. Hamid, you are in LA, correct?
We're in Los Angeles.
Yes, Los Angeles. And Dr. Carvajal, you're gonna have to remind me where you're at now. New York, okay.
New York.
So we've got
Yeah.
both ends of the country here. But there are people in the middle, like Dr. Moser and Dr. McKean. Dr. Moser is in Scottsdale, where I live, and then Dr. McKean is in Tennessee. And if you guys as patients need help finding a doctor in your local area or connecting with someone who you want to connect with virtually, please reach out to us here at A Cure Insight, at contact@acureinsight. And if you're having difficulty finding someone, we can make sure that you're linked up with someone who is well established.
And I think it's, I think, there are a lot of other colleagues who cannot be on this call. Is it just too many people? And they're everywhere.
And it's such a tight community. I mean, I have to say I mean, I think oncology as a field is really close, but this uveal melanoma community, it's we all know each other. We all work really well together.
No, that's such a good point too. And I think the other thing to keep in mind too is that because of all this data that's starting to come out, there's more interest in the oncology research and the oncology realm for doctors, for cutaneous melanoma doctors even, to go, hey, what is this and how can I learn more about it? So I think just the fact that you guys have been so steady in what you do, and then the fact that the data is showing or kind of promoting this further interest in uveal melanoma from other doctors is helpful. But like you guys have said, it's still so important that patients see someone virtually or in person who is a uveal melanoma medical oncologist.
Agreed.
All right. Well, I am not seeing any other questions come through. So if you guys don't have anything else that you wanted to add, I don't know if Dr. Katz, I think he had to step out, but yeah, Dr. Katz had to step out, but Dr. Hamid, Dr. Carvajal, do you have anything you'd like to say to end?
I just recognize A Cure Insight for what you're doing, and then note that your presence is not only welcome, but it has helped this field.
Thank you so
And
much.
I echo that, and the community. This is really important. So thank you for doing this.
Yeah, well, and I'm just one person on the team, guys. There are more people behind the scenes with the board and with Hannah, with A Cure Insight. So we're grateful for all of them who have helped get the word out about this webinar. And we're grateful to you guys both for being here.
One more point to make, that the dissemination of information at this date and time is so quick, not to be overlooked for the patients who are on this call. Please follow the social media for places like A Cure Insight, some of the other melanoma support groups, the Melanoma Research Alliance, Melanoma Research Foundation. These are the people who, disseminate information, and also the physicians who are on this. They have great Twitter presences, and they are available. You can learn a lot about your own state and what's available to you.
No, that's such a powerful thing too. Social media, connecting with social media and not being afraid to utilize that tool. So thank you for drawing attention to that. That's my other realm is the social media. I make sure we've got posts, we've got things. And the other thing I would add is follow the social media for places like Replimune and TriSalus Life Sciences. These different companies that are working on trials or releasing data. You may see data faster than your doctor can get a hold of it, or, you know, because you see it on social media first. And then if you bring that to your attention, to the attention of your doctor in an appointment, then you guys can look at the data together. Because like you guys have said, this is a fast changing field and sometimes there's new updates every couple months. All right. Well, thank you guys both for being here. I'm going to go ahead and excuse you both so you can get to your weekend. But thank you guys for being on live and thank you both for your time.
Take care. Thank you, guys.
All right.
Bye.
Bye. All right. We're going to take just a super short break as we just make sure that Dr. McKean and Dr. Moser are able to get on live. So just bear with us if we have any technical difficulties, but we're just going to let them come on for the next discussion. Hi, Dr. McKean. Hi, Dr. Moser.
Hi, good morning.
Wonderful. Okay. I can hear you guys both fine. And I'm going to actually, just so that I don't mess up the name of your institute where you work at, Dr. McKean, I'm going to let you say where you're from, because I'm like, my brain is, I mean, you know, it's like
That's
7
fine.
o'clock in the morning.
It's early for the
Yeah.
West Coast folks over there.
Yes. It is.
So thank you for inviting me to join you guys this morning. Really excited to be here. So I'm the director of the Melanoma and Skin Cancer Research Program at Sarah Cannon. So I'm at Sarah Cannon Research Institute at Tennessee Oncology. And like you said before, I'm based in Nashville, Tennessee.
Wonderful. Thank you so much. And Dr. Moser is from Scottsdale, Arizona, and he's with Honor Health Research Institute. And I'm going to let you designate anything else you want to explain about what your role is there.
Yeah, so that's exactly correct. So I'm one of the medical oncologists at Honor Health Research Institute, and I lead our skin cancer and uveal melanoma programs. Yep.
Okay. So we have you guys down to talk a little bit about what we shared in our newsworthy notes on ASCO from Kevin, who is one of the people who goes around during ASCO from our medical kind of side of the patient board. And he's really good at just diving into the information and just nitpicking anything and everything from these conferences that has to do with uveal melanoma. So you guys have this list of 12 novel therapies that are related to uveal melanoma, and we're just going to kind of let you go through and talk about those. And then I'll be here to kind of just jump in with conversation as well as I have questions or things like that.
Perfect. So we just have some very basic slides just to kind of, since we're talking about 12 different things, just to kind of help people keep them straight. So as we already said, you know, I'm Justin Moser. My colleague here today is Meredith McKean, and we're just going to walk through the different trials. So the first clinical trial that we're going to talk about is the Plume clinical trial. And this is a clinical trial out of, I think, Institut Curie in France, but definitely somewhere in France, where they're combining pembrolizumab, Keytruda, commonly used immune therapy for both melanoma and uveal melanoma, with a pill called lenvatinib or Lenvima. Lenvatinib or Lenvima is one of these pills that has many different effects, but mainly blocks blood vessels. And this combination of lenvatinib and pembrolizumab has actually been studied in patients with refractory cutaneous melanoma. And the early studies suggest that there is benefit. Because of that, you know, they're looking at this combination in uveal melanoma. This study is currently ongoing. It's only for patients who have previously not seen immune checkpoint inhibitors. For people who are HLA-A*02:01 positive, you could have gotten Kimmtrak or tebentafusp first. And I believe that when they presented at ASCO, they were about 50% accrued. I think they had about 30 of a planned 60 patients. No data has been reported so far with this study, but they are currently enrolling. And it is an option for those who are in France. I do not believe this trial is in the United States.
Great. And I'll just add with the lenvatinib pembro, it's a combination, right? That's been approved for a number of different tumor types. And so now they're kind of looking at it in uveal. So I know one of the most common questions we'll get in the clinic is, what's your experience? What do you guys know about this treatment regimen? And you'll see as we go through, most of these are novel compounds, but some of these we are kind of recycling some drugs that are used maybe in other tumors, or we know some about in uveal. And so that's always a good question to ask, because sometimes even if there isn't a lot of experience in uveal melanoma, well, maybe there's been some experience maybe in other tumors. And I echo Dr. Mosier, like, feel free to type in the chat. Our goal was to keep these slides basic so we could really try to engage in conversation and not overwhelm you with too much information going through, kind of all the highlighted studies for uveal melanoma. So the next study that we are going to highlight was another one of Immunocore studies. So this is F106C. So everyone's familiar now with the FDA-approved Kimmtrak. So this is another bispecific, kind of a similar molecule, but now this is now targeting PRAME. So PRAME is generally expressed in, we consider it a cancer-associated antigen because it really should only be expressed kind of in fetal cells and the testes, because it's involved in meiosis and cell division. But we know there's increased expression in primary tumors that increases the risk for metastasis and increased expression in that metastatic disease. There's been a lot of work done on this with Dr. Harber's group at UT Southwestern. So this is a really common target that we've seen in several studies. But I think this is the most promising one, or certainly the furthest along, unless you can think of another one, Dr. Mosier, that's further along. But I think this is certainly the one maybe with the most data at this point targeting PRAME. So this study, it's a phase 1/2 study. It is enrolling a number of different solid tumors, but they are specifically interested in uveal melanoma patients. Again, patients have to have the HLA-A*02:01 positive status. This is something that can be checked standard of care, but would have to be repeated for trial eligibility. And then this is a frontline study right now. So just important, I think, as we discussed the last hour, some of these studies, one of the important things to know in sequencing is just, this is a study that was previously enrolling patients that were previously treated frontline, but they do have more data, and they presented some of this at ASCO, showing that of the 6 patients that were treatment naive treated up front, 3 of 6 patients had a partial response. And so based on that, they really think probably the best option here is frontline. Patients could have had prior liver-directed therapy, but this is enrolling, I think, certainly a great option. The safety data that's been demonstrated so far, I think overall well tolerated, but I think very similar to what you'd expect from Kimmtrak, because it has a very similar mechanism of action, just a different target. And so it is that weekly dosing. And so looking for a center that's closest to you would certainly be the best idea. But Dr. Mosier, any thoughts on this study? Because I know it's been a while around.
Yeah, no, I completely agree. And, they presented the first part of this study. I think Dr. McKean was part of that. And then Hamid, who just got off, presented it at ESMO last year. And, obviously we don't know, the data is still early and we need to mature. But to me, it looks like it's just as good, if not even slightly better than Kimmtrak. So I think this is a great option. If you're someone who lives around a center that offers this trial and you're debating, Kimmtrak alone or this study, I think this study is a great option given the exciting data that they've already presented last, I think, October or September, whenever it was. Okay, so we'll jump to the next study then. So this is a phase 1 study of a molecule called DYP-688. So this is a drug from Novartis. And DYP-688 is a drug antibody conjugate. Sorry about that. So what that means is it's an antibody directed to a protein that's expressed on a cancer. On the back end of that antibody, they actually attach a payload specifically designed to kill the cancer or, slow the cancer from growing. The benefit from antibody drug conjugates, we think, is that it's a way of selectively delivering, cancer treatments to the cancer with getting less of a dose to the normal tissue. And we think that makes it more effective and also safer with less side effects. And we have a lot of drug antibody conjugates that have been approved for other cancers in the past couple of years. I think starting in the lymphomas, but now in, breast cancer, urothelial cancer, and, lots of different things. And I think that concept is very true. We see good response rates with these drugs with less side effects than chemotherapy. So this particular drug antibody conjugate targets a protein called PMEL17, which is actually one of the things pathologists use to diagnose melanoma under a microscope. If you get a biopsy and they don't know what your cancer is, they will actually stain it for PMEL17. So we know that this is very much expressed on melanoma cells. And the payload is actually a drug that blocks GNAQ and GNA11. So we know for uveal melanoma, about 93 to 96% of these tumors, they are mutated and driven in GNAQ and GNA11. So this is a way of selectively kind of shutting down that mutation. I do have a typo here. It says the status is closed. The status, they actually are still enrolling in the phase 1 escalation right now. So I think this is available at 3 or 4 sites in the United States and more sites across the world. But, drug antibody conjugates have, as a class in other tumors, have shown good response rates, not necessarily durable response rates like immune therapy, but we don't know. This drug could be different. So I think this is a great option if it's available. But again, it is only limited, I think, at 3 or 4 centers in the United States so far. Any thoughts, Dr. McKean?
Yeah, I agree. I mean, I think exciting to see antibody drug conjugates come to uveal melanoma. But yeah, I think right now it's a very pretty limited study, limited sites. So I agree. I think certainly if it's an option, I think certainly worth looking into. But I know it's very limited at this point. All right, so moving on to IDE 196. So this is a drug that's been in development for quite some time. I'd almost say 10 years. It's been a long time coming. And what IDE 196, now called darovasertib, is it's a PKC inhibitor. And the challenge is, so the opportunity here is that is a PKC inhibitor just downstream of GNAQ, GNA11, just like some of the other compounds that we've discussed. And so the idea is that this would be a targeted therapy that should be an option really for most uveal melanoma patients, since we know the GNAQ 11 pathway is activated as kind of a defining feature of uveal melanoma. And so it's nice because it is specific to uveal melanoma, but because those mutations are so prevalent, we don't have to do any pre-screening. We just think most patients would benefit. The challenge has been, historically when this was previously under development, was just trying to manage the toxicity. But yet we know there's been efficacy. And when this drug was under, I believe it was when it was under Novartis, at one of the lowest doses, the patient had a complete response. And so there's been a lot of excitement for this drug. This drug is currently still in a phase 1/2 study. Dr. Moser and I are both involved with this study and will continue to be involved with this molecule. They've moved from investigating this drug as a monotherapy into combination. And that was, back like what Dr. Hamid and Dr. Carvajal had said, the importance of doing those biopsies. We agree with patients, love to avoid biopsies. We understand, especially biopsies of the liver, they are uncomfortable at best. But it was based on, biopsy information that helps the development of these molecules, and being able to understand from liver biopsies that, hey, it looks like there is increased c-Met expression. And so they were able to add on a medication, crizotinib, that was already has already been developed for patients with lung cancer, but does target c-Met, to be able to add that drug into development and to now be able to see really profound responses. So there's been data kind of presented in kind of every 6 months or so by the company. The most recent data was the combination with crizotinib, demonstrating that patients with liver-only disease, I believe it was 11 of those patients, all those patients had some level of tumor shrinkage. And then the response rates in the greater population, about closer to about 40% or so. Now, this drug does come with side effects. And I advise any patient that, hey, here's a list of side effects you need that this drug can cause. You need, we're going to continue to see you closely, manage the, please call us, because there are definitely side effects with this treatment, but we can help you manage them. And certainly over those first 4 to 8 weeks, it's really just trying to figure out how does this medication affect your body. But I think certainly a really great and exciting option in the field. And this will be going into phase 3 in the frontline setting later this year, hopefully. So I think this is another one of those studies that certainly if you can look at this in the frontline setting, I would highly encourage you to do that, because it won't be available in later lines except as compassionate use, and that's a really challenging route. So Dr. Mosier, I know you've been involved in this trial and will continue to be moving forward. So appreciate your thoughts on this.
Yeah, I echo everything you said, and I think, we've had some limited data presented about this study so far, but Dr. McKean's actually going to give us a bigger update in Paris at ESMO later this year. And I think getting that information is going to be really exciting. What they presented so far, like Dr. McKean said, is we do see tumor shrinkage. And I think when you look at the data that was presented by Dr. Katz and Dr. Hamid, a lot of times in uveal melanoma, we don't necessarily see deep rates of shrinkage. With a lot of treatments, we just see kind of stable disease, which is good, right? If a patient's cancer isn't growing and isn't causing them symptoms and they don't have side effects from treatment, that's a win. But, this had, from that, what they reported can cause shrinkage. And so I think this is a really good option, especially for people with bigger tumors, symptomatic tumors, where we really need that shrinkage, because what they presented so far is we can see shrinkage in a lot of patients. And so I think this is a really good option, and I'm excited to see it move forward. So the next study we'll talk about is very similar to the last study we talked about. So this was a phase 1 study of roginalcilib. I'm probably not saying that correctly. So the medication, the IDE 196 or darovasertib that Dr. McKean just talked about, is a PKC inhibitor. So PKC stands for protein kinase C. And protein kinase C, there's actually 9 or 10 different isoforms of this protein, which means, it's not one molecule, it's actually kind of a family of them. And, IDE 196 is a pan-PKC inhibitor. It tends to block all of them. And we're starting to see people get more specific, and this is an example of that. So this drug targets PKC delta, which is one of the 9 family members of PKC. So it's more selectively blocking this isoform. And this is exciting because in general, when we talk about pills to treat cancers, the more specific you can be, the potentially more potent you can be, which means it can be more effective, maybe. But definitely the more selective you are, the less side effects you get. And so we know that PKC is actually, this specific version of the protein is really related to the immune cells and helps kind of suppress the immune system. So this drug really targets that function of protein kinase C and tries to reactivate the immune system. So this was a completed phase 1 study that they did. And initially it was in multiple tumor types. And then when they saw some interesting data in uveal melanoma, they kind of expanded to, I think it was a 20-patient arm of uveal melanoma. So this is an oral pill that you take once a day. I think one of the exciting things about this is their serious side effect rate, which is a grade 3 side effect is what we call that, was only around 7% at their high dose, which is very, very low. You know, there's very few cancer treatments anywhere in any tumor that have a 7% significant side effect rate. When they looked at the, the responses in uveal melanoma, I don't believe they reported tumor shrinkage, but they show that in patients with refractory uveal melanoma that they treated, their median survival was about 20 months. Now, for Kimmtrak, which is our only FDA-approved therapy for uveal melanoma, if we use it first line, we see an average survival of about 21 months. So these are patients who have failed standard therapy and are living just as long on average as Kimmtrak. And so it's a small patient population. So we clearly need to get more, continue to study this to figure it out further. But if we really have a drug that can provide that level of benefit with that little of side effects, with an oral pill that you take daily rather than something you have to come in once a week, I think that's very exciting. And so this study is closed. I believe the company is planning some further options about trying to start a phase 2 or phase 3 study. I don't think we have timelines for that yet. But I think the concept of targeting the specific form of PKC rather than all PKC, given the responses we've seen in the last study Dr. McKean talked about, is exciting. And we're going to start to see some more trials come into this space later this year about targeting other PKC isoforms rather than just Delta. And so I think this is an exciting field. Dr. McKean?
Yeah, I agree. I think this is certainly one of those studies to keep an eye on, because like you said, I think we've got demonstrated track record with this mechanism, and clearly with having a little bit more specificity and having a really nice side effect profile. I think that's certainly something to keep an eye on. All right, so moving on to LNS 8801. So this is a novel mechanism of action. So this is a GPER agonist. And so GPER, so the GPER agonist, decreases c-Myc protein depletion, which increases melanocyte differentiation, right? That's the challenge with cancer. Some of these are kind of baby cells that keep growing and don't become kind of adult melanocytes. And so they just keep growing and dividing. So this is a novel mechanism of action. They've evaluated this for a number of different tumor types, but specifically uveal melanoma, both as a monotherapy by itself and in combination with pembrolizumab or Keytruda. And so we do have some early results showing that actually pretty great toxicity profile. Patients were able to manage side effects very well. And of the 15 patients that were reported with results at this point, some of them had just gotten monotherapy, some in combination. There was a 50% disease control rate. And so what that means, and I guess trying to decipher when you're looking through some of the data from these conferences and trials, disease control rate includes patients with stable disease. And we define stable disease as patients that have had up to 30% growth or 20%, 30% shrinkage or 20% growth. And so it's actually a fairly large range, but it's still clinically important. And so this drug did show a 50% disease control rate, and did have one patient that had a greater than 30% shrinkage of their tumors consistent with a partial response. So I think really exciting to see this mechanism of action. I think this one is still enrolling, but I think that's certainly something on clinicaltrials.gov. If interested in, say, a trial like this, if you go on there, it'll show the different sites, and they should have a contact person that you can reach out to say, hey, if I'm interested, what, is this still enrolling? And there's eligibility criteria listed there. But I think certainly an exciting, different mechanism of action, and something that we'll be watching closely. Dr. Moser, any thoughts that you've had on this trial?
Yeah, I agree. When it just kind of a bigger picture of this study, it's exciting because the class of proteins called G-coupled protein receptors have notoriously been something that we've not been able to figure out how to target with drugs. You know, we target tyrosine kinases, which just is another type of protein, all the time. And pretty much all of the pills you take for cancer are tyrosine kinase inhibitors. But we've really struggled to build therapies that can block G-coupled protein receptors. And we know GNAQ and GNA11 are G-coupled protein receptors. So the fact that we're starting to see people cracking how we can target these things, I think is really exciting for the future, because this is a brand new field that no one has been able to do before. And, you know, the downstream ramifications of being able to do this, I think, are really exciting. Now, we probably won't see this for 5-10 years, but it's really exciting new science. I realized I had the questions thing closed, and there was a couple of questions about the trials we already talked about. So we'll go back and address them. So for DYP688, the question was, is this a systemic option and not a liver-directed therapy? It is. Most antibody drug conjugates, and I believe this one too, is an IV medication that goes everywhere. So it is a systemic option. And then someone else asked, why was PKC delta chosen as the target rather than the other 8? I think the reason for that is PKC delta is the most notorious isoform of PKC that suppresses the immune system. And they were really trying to target a drug, or trying to make the drug have immune effects. I believe. This is me guessing. I don't know for sure. But I believe that's why they chose that one, was because that is the immune effector PKC isoform.
So Dr. Mauser, can I jump in?
Yep.
Oh, I'm echoing. Can I jump in with a couple questions kind of around a few of the things that you've been talking about?
Absolutely.
So you guys have talked about obviously a few different things that are systemic therapies. So like as a patient with uveal melanoma myself, one of the things that like I keep hearing more and more in the metastatic community is that it's vital, at least as early as possible. That's why we screen so often, that it's vital to try to like get disease control in the liver first. So do we know if there's any data on these systemic treatments, these ones where it's just a pill or it's an IV, that shows that it's successful in containing liver disease as opposed to whole body disease? Because I think that one of the challenges that I see in the metastatic community is when a patient who has only rampant liver disease is trying a systemic trial that has not yet been proven effective across the board in the liver. And I just feel like there's got to be a balance between how do we figure out the best time to do liver directed and the best time to add in a systemic treatment.
Do you want to go ahead with that, Dr. McKean?
Sure, I can get started. I mean, it's a great question. And I think ultimately, I think those of us in the field, there's kind of differing opinions. I think most folks agree there's certainly a role for both liver-directed and systemic therapy. And I think certainly as we're getting more uveal melanoma specific drugs, we are able to offer those a little bit earlier. Now, when to your question about patient response and achieving response in the liver, so for a trial, the goal is that you have to pick representative lesions for that patient's disease to be able to follow, and you're doing specific measurements. But even if a patient has disease in the lungs and you're following those lung nodules and those have shrunk, but the liver disease is growing, that would still in general be considered, they'll do the measurements, but then you also have to have your clinical acumen to say, hey, is this actually progressing? And so when these trials present saying a partial response, that has to take into account their disease diffusely. So I think there are some studies, so like the IDE 196 darovasertib trial, they've done the work to show that the liver metastases showed increased c-Met expression on monotherapy, and that's why they added the crizotinib. I think we are getting smarter about trying to understand the liver metastases specifically, but I do think some of these trials are maybe a little early yet for them to really be able to say, we're clearly seeing, disease shrinkage specifically in the liver. But that's where, they're able to say, hey, does it look like the disease diffusely is overall decreasing? Dr. Mosier, I know you have a lot of experience as well in, early phase trials where most of these are lying.
Yeah, no, I agree. And I think, when we have studies, I think you explained the response rate perfectly. And when we have studies that have reported responses like the IDEA drug, we can take that to mean that they had responses in their livers, and so many patients with uveal melanoma have liver responses. So, if we have something that causes shrinkage in a substantial number of patients, I would consider that probably equivalent to a liver-directed therapy, because it probably is going to cause shrinkage in the liver and give liver control. My personal approach is, if you have moderate or higher disease burden, I try to do a liver-directed therapy first, try to get that back under control. But I do like to start you on a systemic therapy or a clinical trial, if that's the best option, quickly soon after that, because, the liver-directed therapies, other than, percutaneous hepatic perfusion, which is currently being reviewed by the FDA with Delcath, they definitely don't control disease outside the liver, and their durability may not be that great. So I like to start something systemic too, to try to give it that kind of holistic approach, for lack of a better word, about targeting the liver, but then also trying to do something that has the potential, although maybe small, to provide that long-term durable benefit.
So can I ask a question about kind of what you guys mentioned about the data around, for example, if someone is on a trial and they have lung nodules that are shrinking, or maybe bone nodules that are shrinking, but they have liver metastases that have grown. Can you just kind of maybe better explain how that data kind of equals only a 10% progression instead of what looks like a 50% progression, or whatever the math turns out to be? Because I think that sometimes that can be confusing for patients to understand. Sometimes there might be shrinkage somewhere and growth somewhere else, and they kind of cancel each other out in the data.
Dr. Mosier, do you want to start with this one, or you want me to?
Sure. It's extraordinarily confusing, and a lot of times the only way because we do exactly what Dr. McKean said, which is we pick these lesions and then we decide which ones we're going to follow, which ones we're going to follow but not measure, and then we report a number. But there are some patients out there where you look at their report and you say, oh, well, if I would have just randomly chosen lesions differently 3 months ago when I put them on the study, it would be a different result. And I think that's just the imperfect nature that we have in terms of following patients and trying to quantify a response, because personal benefit is such a qualitative thing. It's really hard to quantify that. So yeah, it's very hard. And sometimes even as investigators, when we're looking at data, we can't even necessarily know that either, just based on how we do it. It's definitely something it would be great if we had a better option, but right now we just don't.
Well, and I'll add to that, most of these trials, there's, any of them, there's a protocol and you have to stay within those guardrails. But say if you do meet progressive disease, like based on the lesions that were selected at baseline, there's greater than 20%. Most of these protocols will allow you to, if you and the patient decide to stay on, oftentimes they'll say, okay, we'll let you stay on for another 4 to 8 weeks with another scan just to see, because sometimes for patients with uveal melanoma, we can see some growth, but then things remain stable again for a period of time. So I think it's worth recognizing that the protocols have guardrails, but there is still some flexibility for just doing what's best for you as a patient. So if that is, hey, should we try to give it a little bit more time, but if we're still seeing growth, then okay, this clearly isn't working. Or to say, hey, even if there's only been slight growth, like you're feeling much worse, like just, and we don't think it's side effects that we've been able to manage, then let's just come off study. There is that, there is still some flexibility that you can, like you and your doctor can decide what's best within the trial. Now, certainly you want to be able to give that drug an opportunity to work, right? So, if you're feeling miserable at 2 weeks on, you want to try to say, okay, is this toxic? Like, what can we do to help with side effects if this is from the disease or from the treatment? But I think one of the things I'll talk to patients about is, like, there are these parameters, but like Dr. Moser said, if the lesions that you picked, that all the other ones seem to be shrinking, well, then that's something that you go back to the sponsor and say, look, we think there's clinical benefit. We want to try to stay on a little bit longer. That there is opportunity for that on a trial.
I think that's reassuring for sure, because it's important, I think, to note that, like you said, the studies are trying to quantify something that really is more qualitative data. It's more like looking at quality of life, looking at how long is the patient able to continue survival without, significant progression. And that those things are of far more value than did everything shrink a ton. Because like you guys have all talked about between this session and the last session, sometimes that shrinkage takes a really, really long time, if it even happens at all. There's some patients who get stable and then they stay stable for years and there's no shrinkage, but their tumors appear to be dead, or they appear to just not be growing, or whatever is happening. And it's so individual to every person. So I like what you said about just having that discussion with your doctor about, what's the risk benefit of going on or off the trial. And is it time to move on, or is there room for pushing to continue?
And I'll add, a lot of these studies are early in development. So sometimes it's worth asking on these trials, sometimes if it's dose escalation, and a patient went in at, say, dose level 2 and the study's at dose level 4, then a patient can what we call intra-patient dose escalate. So you can reach out to the sponsor and say, hey, this scan has shown some growth, but this patient's tolerating the treatment well. Is there opportunity to go up in the dose? Sometimes some of these targeted therapies, they are exploring adding in another medication at progression. So I think it's certainly worth kind of going back, and we're Dr. Moser and I, we're always kind of going back to the sponsor to say, what else is there that we can do to try to, maximize this patient's benefit, and try to keep them on trial if you think there is an opportunity to try to, intensify treatment someplace.
Okay.
So we'll go on to the next study then. So the next one that we'll talk about is a phase 1b study of SLC45A2 endogenous T cells. So this is a study that was done at MD Anderson in Houston. And what they did is they took blood cells from patients' peripheral blood. So they put, big IVs into these patients and then removed their immune cells. They only included patients with certain HLA types, and that's HLA A0201 or HLA 2402. And they took the immune cells out of the blood, and they didn't provide the exact information from their poster that they presented, but they trained these cells to attack or to express, to attack or recognize, I should say, a protein called SLC45A2. And so this specific protein is actually like a melanosome transport protein. Melanomas are full of melanin, right? That's what makes them pigmented. So melanosomes are just the little blebs in the cell that contain melanin. So that means that this protein is really specific to melanoma cells and not to normal cells, which means, in theory, we should be able to direct an immune system response to cells expressing this protein without having a lot of side effects because it's not expressed on normal tissue. I don't believe these were genetically modified cells. I think they were just trained or selected to recognize this protein. Once the cells were created, patients then received, I think, 2 to 3 days of light chemotherapy to make room in the bone marrow for new cells. Then they were given the cell infusion either through an IV or through the hepatic arterial infusion, similar to what Dr. Katz talked about with the TriSalus system, except they didn't use, I don't believe they used that little pressure pump he talked about. And then after this, patients got low-dose interleukin-2. So interleukin-2 is a cytokine that tells T cells to grow and expand. And usually when we do cell therapies, but not always, we give interleukin-2 afterwards to try to help the cells that we give grow and expand and become more numerous. So they, I think they treated, can't remember how many people they treated. I think it may have been about 20. They didn't notice any responses on this trial, but there was some immune changes in some patients with stable disease. I think their median progression-free survival was on the order of about 6 months, if I recall correctly. They did note that they get better immune cell in expansion when the cells were given IV rather than infused into the hepatic artery, and the causes of that aren't clear. But currently based on clinicaltrials.gov, this is not actively recruiting, but it is interesting studies. Anytime we see cell therapies and other tumors have been very potent, and it really changed the landscape of blood tumors such as lymphoma and leukemia. So we always get excited about trying to use cell therapies in melanoma and other solid tumors as well. So I think this was a brilliant study to do. Hopefully they're looking at ways to kind of tweak it and make it better, and we'll bring it back. Any other thoughts, Dr. McKean?
Yeah, no, I agree. I mean, such a just amazing technology and potential option for patients. And so I think certainly something to kind of stay tuned about to see, where this goes in the future. All right, so PRT-811, so this is a PRMT5 inhibitor. So there's actually a number of PRMT5 inhibitors in development, but this was one where they were specifically interested in looking at patients with uveal melanoma. So this same company, Prelude, had a prior PRMT5 inhibitor, PRT-543, wasn't as well tolerated, but there were some early signals. This was kind of their second generation PRMT5 inhibitor. And this was specifically of interest for patients with splice site mutations. And so we know for patients with uveal melanoma, it's actually now showing up for Castle on the 7-gene panel with a primary tumor. You can see that this is one of the intermediate risk factors for patients with a primary tumor. So we know this is kind of dysregulated RNA splicing. And so a PRMT5 inhibitor, the goal is to basically try to correct that with it's a methyltransferase. So basically trying to tell the cells not to splice the RNA exactly where they are. So this was a study that was ongoing for several years. They did enroll, they saw some early signals in the dose escalation. So they did enroll 10 additional patients with a splicing site mutation and then 13 without, because they weren't sure if it was necessarily specific to patients that have an SF3B1 mutation. And so, what they saw is that, so this was a brain penetrant molecule, which is nice. We know the rate and risk of brain metastases for patients with uveal melanoma early in disease is low, but we can start seeing some of these brain metastases later in the course of a patient's disease. But in general, it was well tolerated. The side effects were primarily GI, nausea, vomiting, diarrhea, fatigue. We could see a rash or thinning of the hair. But what they showed was that there was about a 50% disease control rate, including several patients with tumor shrinkage. This kind of gets back to our discussion before. Tumor shrinkage, so prolonged stable disease, but those patients had about 20% shrinkage as opposed to meeting that threshold of 30% shrinkage. But there was one patient with a confirmed PR. So this study is currently closed, but I think there's still more for us to learn in this space. And so I do think, there were certainly some patients that benefited, but it wasn't a home run, that it's ideal when you have like a specific marker for a patient, right? And you can match it up with this drug specifically should work for you. There was a signal there, but they closed because they weren't seeing consistent benefit. And so I think more to come here in this space. for patients, because that's what we would love, is to be able to say, as much progress as we've made for patients with uveal melanoma by identifying you have a BAP1, you have an SF3B1, like how can we in turn help that, with our treatment selection.
I agree. And I think, if we can find something to target the specific mutations like the SF3B1 mutation, which we see in a handful of uveal melanomas, and we know actually means if you have a primary melanoma and you have a mutation in FS3B1, you're actually higher risk for a very late recurrence. A lot of times when we send capsule testing, we give people their 5-year quote. But for these patients, we typically see, recurrences much later, 10, sometimes 15 years later. And so I think if we can, kind of revive this space, like Dr. McKean said, and really learn more about how can we target this specific mutation and exploit it, there definitely is benefit to the community. So hopefully this isn't the last thing that we see in this space. The next thing we'll talk about is tumor-infiltrating lymphocytes, and we're going to kind of talk about this broadly because there's actually multiple different studies in different sites going on, one of which was presented at ASCO this year. So first of all, what are tumor-infiltrating lymphocytes? So lymphocytes are just the immune cells that attack, bacteria, viruses, different infections, but also attack our cancers. And tumor-infiltrating lymphocytes just means they're lymphocytes that are in the tumor. And it's been well known for, 50 plus years that when pathologists look at cancer under a microscope, the cancers that have a lot of lymphocytes inside tended to do better. And so because of that, starting, I think, the late 70s, the National Cancer Institute and the NIH surgical branch started doing tumor-infiltrating lymphocyte therapy, where they would cut out these tumors, break the tumor tissue down into a single cell suspension, remove the lymphocytes, and then grow them from a couple hundred to a couple billion. They then give these back to patients, and this has been shown to be beneficial. And currently, there's been a couple trials from Iovance that have shown benefit in patients with cutaneous melanoma, and this is actually currently being reviewed by the FDA. I think the FDA is supposed to give a response by late November. But we're also studying this in uveal melanoma. So I believe UPMC in Pittsburgh has a TIL trial that's actively going. There's a trial at MD Anderson that we'll talk about that was discussed at ASCO. And then Turnstone Bio actually has a trial of their TIL therapy that's going on at kind of multiple sites. So when we do tumor-infiltrating lymphocyte therapy, it's very similar to the last therapy we talked about, where you give people chemotherapy for a couple of days. Usually in this case, it's 5 to 7 days. We give the cells back, and then instead of giving low-dose IL-2, the cytokine that helps the immune cells expand, they give high-dose IL-2, which has to be done in the hospital under monitoring, because it can be done safely, but it can be very toxic if, not done well or if you get side effects. So in the study that they presented at MD Anderson, they successfully collected and grew TILs from, I think, 30 or 40 plus patients with uveal melanoma. 9 patients were treated with this regimen. Of the 9 patients that were treated, they did see 2 responses, so a 22% response rate. The exciting thing about this is both of those patients who responded continued to have that response well over a year. I think one patient was 16 months and another patient was over 20 months. So this is a very intense but one-time therapy that can provide durable responses. And that durability we see in the other TIL trials that have been studied both in cutaneous melanoma and that they're looking at in other tumors currently. So currently these studies are recruiting. There's just very limited availability between all 3 studies. And currently I think it's only offered at maybe 5 or 6, 7 sites in the US. So it's very limited. There tends to be long wait lists. But if this is FDA approved for melanoma later this year, uveal melanoma does fall under the umbrella of melanoma. And so if we continue to see good results with this, it is possible that this could be done as standard of care if it's FDA approved later this year once the centers get up and running. So I think this is an exciting thing to kind of follow. Again, it's very intense. It's very toxic. It's about a month of treatment. And so you have to be pretty fit to get this. but it is a one-time treatment that can be durable, but a very intense one-time treatment.
Yeah, I think certainly an exciting area, and like you said, it's been in development for a long time, because it is an intense treatment, but would really love to be able to see this as a more common option for a highly selected patient population. I think cutaneous melanoma, sometimes it's easier to get larger tumors, but especially for patients with uveal melanoma that maybe has, a cutaneous metastasis, as opposed to having to do multiple cores of the liver. I think I agree with you. I'd love to see a little bit more availability, because right now it is just a challenge, just being in a couple centers, waiting lists, and sometimes you don't have the time to necessarily wait just to see if you're a candidate. Great. And I think we're rounding third, headed to home here with our trials list. So briefly, just going over the Scandium and Scandium 2 trials. So these were trials that were presented recently at ASCO out of Sweden, some of our colleagues there. And so these are trials that I think the important point was just, as we look at maybe some combination therapies, what does that look like in the field? So these were patients, the Scandium 1 was just doing high dose melphalan in the liver. Scandium 2 was with ipi/nivo. And basically they showed that there could be some significant toxicities. There was one patient that wasn't able to proceed with liver-directed treatment after getting neoadjuvant treatment. But they did show that of the patients treated, there was an overall response rate of 63% in arm A, and that was with the ipi/nivo. There were 3 complete responses, 4 patients with a partial response. So with the treatment alone was 24%, and then you add in that ipi/nivo 63%. So I think, Dr. Mosier, you're probably like me, you get a lot of questions, like you were saying, with liver directed, you like to follow with systemic therapy. But a lot of folks that will say, gosh, can you just do the ipi/nivo at the same time? And I think there's a lot of hesitation just with doing too much to the liver, right? We know we want to be aggressive with the liver, but also we know just with ipi/nivo, you can get immune-related hepatitis. When we're doing that intense liver-directed treatment, can also certainly have irritation of the liver, pain. And so I think what this study was exciting, because it did show that being able to, the goal of trying to control the liver aggressively while doing systemic therapy, I think is going to be an avenue that we continue to pursue moving forward. But I think certainly having to be very, very mindful of the toxicities and how to best navigate that moving forward.
I agree. I think, this isolated liver perfusion or percutaneous hepatic perfusion, the response rates that we see in the liver, especially in combination with ipi/nivo, are exciting. Again, it's very toxic. It's a recurrent treatment, so it has to be selected for the right patient. But I'm excited to see this kind of come out further. I know Delcap, their version in the United States, is currently under FDA review. Hopefully that gets approved and we can start seeing this being offered at more places, because I think for the right person, this can be a really great option. So we got a couple of questions. So before we go to the last trial, we'll answer some questions. So one person asked, where are these infiltration sites? So for the tumor infiltrating lymphocytes, it's UPMC in Pittsburgh, MD Anderson in Houston. And then for the Turnstone Bio study, you can look on clinicaltrials.gov, but the 2 that I know of off the top of my head are Moffitt in Tampa, and then Providence Health in Portland. There's probably a couple others too. I don't know, Meredith, if you know of any others, but those are the ones I know off the top of my head.
Yeah, I think those are the main sites. And, just in case the question was more, like where the infiltration sites of the lymphocytes and the tumor, in case that was also the question, they're really just looking at an easily accessible tumor. And so that's why if there is a, lymph node that's easily accessible, or a subcutaneous lesion that they could, they're looking for something that they could surgically resect to then take out the lymphocytes that have infiltrated into the tumor. And so if they need to, they can do like multiple core biopsies of a liver lesion, but it can be challenging. And that's part of the consultation that they would discuss is like, is there enough tumor that they can reach to try to get those lymphocytes out of the inside? So and either way, you know, those would be the sites where they're doing this, and then the patient eligibility. You know, they're looking. That's why they look at your imaging so closely, and your candidacy, like whether or not you can do TIL. That can evolve over the course of your disease.
So we do have a question here, and I'm going to try and generalize the question a little bit more. But what is, I guess, in your guys' opinion, what's the risk benefit of doing something like immunoembolization at Jefferson that is a little bit more maybe well known in the metastatic treatment community, versus doing one of these trials that's significantly progressed, like tebentafusp mixed with PRAME? I think you guys had mentioned like that Tebi PRAME combination. You guys thought the data looked more promising even than just Kimmtrak alone. But then there's also kind of that balance of disease control in the liver, which immunoembolization can provide. So do you guys have any suggestions or comments there to that kind of a situation?
Yeah, I tend to tell patients, you know, I'll tell them honestly, like, this is a new trial and we need more information, so I wouldn't jump to it right away. Or we have data about this study and it looks exciting. I think for the tebentafusp and PRAME trial, you know, we have data already. We've seen responses. It's a good option. So when I'm trying to counsel a patient between one or two options, it's, you know, we've never compared these things head to head. So we don't know which one is more effective. We just know that they both can be effective. So the conversation I typically have is with side effects and quality of life. You know, obviously embolizations are one or two, maybe three treatments, depending on which type you're getting, and then you're done. You know, versus a study like the Immunocore studies, which are weekly treatments, or other studies where you have to come in every 2 weeks. And so it's really, you know, what's most important to you? Is a one and done treatment and having freedom more important? Is going on a clinical trial that you know you can only get first line and never get again, that more important? And it's really just the kind of holistic picture of benefits, side effects, quality of life, and how much you're tethered to the clinics, and really trying to make the right decision for the patient.
Yeah, I echo that. I do think, you know, some of these studies, I think, as Dr. Mosier mentioned, it is important to understand just like the IDEA study, the PRAME study, they are moving to the frontline setting. And so that's going to be the opportunity to get it. It used to be, hey, at some point you can come and get these treatments, but they are being more selective. Now the PRAME study is still currently allowing liver-directed treatment that you can go on. So, you know, not knowing the specifics of your situation, if it's immunoembolization, that's usually an option that they would offer with kind of a little bit lower volume, less aggressive disease. And so, but I do think it's helpful just to make sure your whole care team knows what your options are, because everyone has a little bit of expertise. They might be able to say, you know, hey, the disease in the liver looks like, you know, from that last scan to this scan, it is advancing a little bit more aggressively. We should probably go forward with liver-directed therapy. And then you can still hopefully go back to PRAME potentially, or, you know, if you're close to the Angeles Clinic, it's right down the street. I don't know if you can be right down the street in easy traffic in LA, but if it is easier to get to the clinic, you know, maybe that is more feasible, and, you know, the data looks really exciting. But I think one thing I try to stress to patients is there's no one right answer, and whatever the results are, you know, presented, we try our best to individualize it for you, but each patient's disease and situation is unique, and we all will try to help you work through that, but there isn't kind of one right answer for every patient.
Yeah, I think that's such an important thing. And I think that really kind of also answers the last question that we have as we're ending. And it's just, how do you decide between how many, like all of these promising trials? And it really just comes down to, it is such an individual discussion with your doctor. And it really depends on a lot of factors for you as the patient that you have to discuss with your doctor openly, like anything from travel and financial feasibility to the potential, response rate and the risk benefit of the various different types of trials. Like, I think what I was hearing from things like the TIL therapy is that maybe that would be something that someone in the younger patient population might be a little bit more likely to be successful with, versus someone who is maybe a little bit older, maybe over the age of 50, they just may not have the physical stamina for something like that. So taking all of those into account, I think is something that when you have a good uveal melanoma specialist who, like we've talked about, who is well-versed in the field, who's consulting with all of the other doctors in the field and just comparing notes, they will help, that will help to make that decision.
So we'll jump to our last study then before we pass it off to Dr. Dalvin. So the last study we're going to talk about is targeted alpha particle radiotherapy for uveal melanoma. So this is one of these trials of this new wave that we're seeing in oncology, which I call liquid radiation just for simplicity. So classically, when we give radiation, you go to a room, there's a machine that goes around you, and there's a beam that goes from the outside to the inside. We now are able to give radioactive particles through an IV, and through different methods, we target them to certain proteins and cells. And so it's a way of kind of giving radioactive molecules IV, but having them self-target to cancers. So we can radiate cancer everywhere in the body versus just one or two spots or targeted spots using the conventional outside-in approach. And so when we think about these liquid radiations, there are two currently approved liquid radiations for the treatment of cancer. One is called they're both lutetium based, which is the radioactive molecule. One is called Lutathera, one is called Pluvicto, and those are for neuroendocrine tumors or prostate cancer. And lutetium is what we call a beta emitter, and alpha and beta are just different types of radiation. And the biggest difference is how far they go. So lutetium is a beta emitter, so it releases radiation a longer distance than alpha particles. And so there's a lot of excitement right now about trying to use alpha emitters because they radiate in such a shorter area, which means we hopefully can see less side effects with these treatments, because if we take that radioactive material, truly stick it to the cancer, and the radiation only goes as far as the cancer but no further, we should be able to treat these cancers with less side effects. And there's a couple of different trials in this space, but this is one specific for uveal melanoma. It's currently in phase 1 studies, I believe, and I believe it's only available at Moffitt. It is for patients with refractory uveal melanoma, so patients with at least one prior line of therapy. And I don't know the specifics, but clinicaltrials.gov didn't clarify if that had to be a systemic therapy or liver-directed, or it could be a liver-directed therapy. And it is currently enrolling. Typically when you do these studies and you get the IV infusion, there are some lifestyle changes you have to temporarily make. All these radioactive molecules are cleared from the kidney. So usually you have to kind of use your own bathroom for a couple of days, make sure you drink lots of water to flush it out. They usually tell you that you should sleep in a different bed from your partner for at least 3 days, and a lot of other things like double flush the toilet and stuff like that. So it's an easy treatment to do, but there are a lot of lifestyle things that you have to be able to accommodate, so you're not inadvertently, giving someone else radiation by sleeping next to them. But I do think, radioligands or these particle radiotherapies are going to be a huge wave in oncology in the next 10 to 15 years. And I'm excited that we're looking at them in uveal melanoma so early.
Yeah, I think this is just an exciting mechanism of action, and I think just a very different treatment than what's available with some of the other systemic therapies. So I agree. I'm excited, and we'll kind of see where this leads. But I think a great option for patients.
I love this. This has been a lot of information. So those of you guys listening, feel free to come back, listen to the recording. This is being recorded, and the recording will be up on YouTube as well as our Facebook immediately following. And then it'll come up on the podcast after our audio is edited. But I don't see any other specific questions. I just want to thank you both for your time. Thank you, Dr. Moser, for joining me in the early bird situation here. It's been fun. But it's just such valuable information. So thank you both.
Great, thank you. And thanks to Kevin. I know we both had looked at, he does like a fantastic summary that's available through your group. So I think certainly stay tuned to Kevin for future research and conference updates. But thanks for having us.
Yeah, thank you, Dr. McKean. All right, I think we're about ready to move on to having Dr. Dalvin join us. Thank you so much for being here, Dr. Dalvin, and thanks for your patience.
Yeah, thanks for having me.
All right, I'm gonna see. There we go. I think that we were having a tiny bit of a technical glitch and I didn't know how to fix it. So hopefully it's fixed. So Dr. Dalvin is joining us, and I apologize that I didn't explain this at the beginning. So for those of you who are still here with us, we are having the Eye on Research mini seminar to talk about the data that was presented at various different conventions, specifically at ASCO, which is the American Society for Clinical Oncology. And there's also the AACR convention that happens, and that took place, I believe, in March of this year, and that stands for the American Association for Cancer Research. And then the last one that we have Dr. Dalvin talking about specifically is the ARVO, which is the Association for Research in Vision and Ophthalmology. So she's our eye expert. You guys have heard her talk a few times, and so we're so glad that she's back here to talk about some of the data that's been released from Castle, some of the data that's been released generally in the eye treatment sphere. And of course, she has a lot of knowledge and expertise in there. So thank you for being here, Dr. Dalvin.
Yeah, thank you so much for having me. I'm really excited to talk about the ARVO data. So ARVO is a really special meeting to me. It's actually the first scientific meeting that I ever attended. And it really has a different vibe than a lot of other scientific meetings. So we have a lot of formal clinical conferences where you typically go in a suit. Way back in the day, we used to always have this meeting in Florida and people would show up in shorts and like a Hawaiian t-shirt. And so it's very welcoming when you're at a new training stage, and it's very international. So this meeting has really wonderful opportunities for building collaborations and for somebody who's newer to really connect with more senior researchers. And so I've become more involved in ARVO over the years, and I'm currently on the planning committee. So we had a really fantastic and very busy ARVO this year, and we have all these different subsections. So our subsection is the anatomy, pathology, and oncology section. And we usually have like a whole poster session and a whole talk session dedicated to uveal melanoma because it's such an important topic for so many of us here. So there were a lot of exciting things presented at this year's ARVO meeting. And as you alluded to, one of the things that was presented was an update from Castle. And they have really done just a tremendous job creating a fantastic prognostic test that we use now very routinely in our clinical practice that probably many of you on the call perhaps have had. And so Katz from Castle, who does just a phenomenal job, presented the updated data on 1,996 tumors and really showed what a great test this is. You don't need a lot of material from this test. It is a direct into the tumor biopsy, but even with a really small sample, they're getting success rates greater than 97% of the time they're able to give out a result. And many of you may know that result typically comes in the form of giving a class status that helps predict metastatic risk of uveal melanoma, which we've been talking about, I think, with some of the other trials today and how that plays into clinical trial eligibility. And interestingly, what they presented was actually, I thought, pretty good news. Of all of the tumors they tested, almost half of them actually came back as a class 1A result, which is the low risk for metastatic disease group. Sometimes this biopsy information can provide a tremendous amount of reassurance. And so for people who get that class 1A, it can really be a big relief and can make a big difference in quality of life. On the other side, they found almost 20% were a class 1B, which is a little higher risk, and then a little over 30% fell into that class 2 category. And that class 2 category is really what's going to want to make us screen, do systemic scans much more often, and potentially think about enrolling in some of these clinical trials for prevention of metastatic risk. Some of the other things that they talked about is how often do they see PRAME positive status in tumors. And as you may know, and they may have mentioned earlier, PRAME expression is a poor prognostic marker. If you have positive PRAME expression, it does put you at higher risk for metastasis. But again, pretty good news, really less than 30%, less than a third of tumors had positive PRAME expression. So actually most of the tumors that are being biopsied may fall into more favorable prognostic categories. And then we know now that the Castle test looks at some sequencing, so it looks at specific mutations. And this is really, I think, going to play long-term into clinical trials and personalizing medicine for patients with uveal melanoma. So they're able to tell us how many patients have a GNA11 or a GNAQ mutation, which are our primary drivers of uveal melanoma. But then they can also tell us about 30% of patients have a BAP1 mutation, about 12% have that SF3B1 mutation that Dr. Mosier was talking about, and about 20% have an EIF1AX mutation. And so these things ultimately, I think when we know a little bit more and when we learn perhaps which mutations play into a drug response, are really going to drive what trials we send our patients to and how we manage people long term. So, the folks at Castle, I think, have just done such a phenomenal job of really creating this information that it not only is helping us right now, but I think is going to play an even bigger and bigger role as we move on to the future.
Well, and to me, this sounds, I know we talked about this back in like January, but to me, this sounds right in line with everything you're working on with the Living Library at the Mayo Clinic. So for those of you who have not heard that episode or that webinar that Dr. Dalvin did, go listen. Back in January, we did a Living Library discussion. She presented on what this is that she's gathering data for. But essentially, it's going to allow us to do just that in the field, where we can look at these prognostic tests, we can look at the makeup of the tumor, and then we can then individualize the therapies that patients are recommended first based on how do they respond in the Living Library. So that's like a super concise version of how this all works. But it's really exciting stuff to see that this individualization of future treatment is coming.
Yeah, I think it's fantastic. And people are working on other ways to get this information as well. So one of the really cool things that they presented at ARVO, Dr. Jesse Berry and Leah Hsu from Los Angeles showcased their work on aqueous biopsy. And they've been working on this for a long time in the retinoblastoma space, but they're trying to bring this into our uveal melanoma patients. And so as we talked about the Castle test, it needs such a small amount of tissue, but it is an invasive biopsy that puts a needle into the tumor. And so we're actually finding that in things like the aqueous humor, which is the fluid that fills the front of your eye, or even in the vitreous fluid in the back of the eye, or even potentially in the bloodstream, we can pick up some of these markers. So they presented that they can look in the aqueous humor and actually find a lot of markers that correlate with what that Castle test result would be. So down the line, we're looking into potentially things like an eye fluid test or a blood test that would be less invasive for people who have a really small tumor, or a tumor that's in your center of vision where you maybe don't want to stick a needle because you're worried about complications like retinal detachment or bleeding that causes scarring and vision loss. Then you may have this more accessible way to get a fluid test. And it could even expand into groups who are looking at things in the blood. Can we detect changes in expression of these markers in the blood over time? So can we actually pick up metastasis or cancer spread before the scans can see it by looking at these markers in the blood? So I think a lot of these things really are going to be on the horizon and are going to feed so much into what our medical oncologists are able to do.
Yeah, and that's super powerful. Just that idea that we can go from the, okay, we have it in the eye, or, we can predict that it's going to metastasize based on these patterns, based on this blood work. And I think what Dr. Moser and even Dr. Hamid and Dr. Carvajal talked about earlier is just this idea of liquid biopsies and those circulating biopsies where we get more information and sometimes more accurate information before it shows up on the scans. And obviously that would be the ideal situation for everyone, is let's know what's coming before it shows up.
Exactly. So, we had so much opportunity. We've got a lot of groups, both in the oncology and the ophthalmology space, who are really working together on developing these tests. So as you mentioned, our group, and there are some other groups too who are working on these 3D models. So just as we've been building our living library, trying to grow individual patient tumor samples, at ARVO, there is a group, Solange Landreville from Canada, who presented a 3D model where they're taking not only the tumor itself, but if they'll mimic an eye tumor by taking surrounding eye tissue, or they'll try to mimic a liver metastasis by taking some of the surrounding liver tissue and letting that organize into its own 3-dimensional structure. And that's really cool, because not only are you getting what is the tumor behavior, but you're able to see then in the lab what's the interaction of the tumor cells with the other normal organ tissues. So these things, we're all kind of on the same page, really excited about 3-dimensional models for testing of drugs in the lab. And these are really cool because, unlike animals, which are very expensive and can be kind of ethically ambiguous and challenging, and unlike 2-dimensional cell culture, which may not really simulate what's going in the body, these 3-dimensional models really are able to predict what's going to happen in a patient in terms of drug response. And there have been people who've used these models for other types of cancers that have been very difficult to treat. And they've actually been able to show that how the 3D model responds to any given drug in the lab correlates really well with how a patient is actually going to respond to that drug in real life. And so what I'm trying to do, and some of my other colleagues have been trying to do, is can we take that same strategy then and take your tumor sample and have a panel of drugs that we might want to test that we know are clinically available, and then know what your tumor responds to in the lab and actually give you the best responding drug in real life. And so I think that's part of where we may be heading. That's going to, again, just personalize our treatments.
No, I love that. So I do have a question around this. When you take like an eye tumor, is there any data that's showing that the eye tumor that, say, then spreads and becomes metastatic in the liver, despite treatment and whatever has happened, enucleation, radiation, proton beam therapy, whatever has happened to the eye, the tumor has then metastasized later in the future? Is there data showing the comparison between the primary tumor and the liver tumor, say, to show that, like, are they different? Are they related? Like cousins, maybe? Are they siblings? Is it like the mom and then the dad, or the mom and then the son? Like, what is the relationship here, and how can they be different?
Yeah, this is a great question. So there are some good data to show that they don't really quite behave the same. And why is that? Is it because they're in a different environment? Is it the selection process of what cells like to grow in the liver or like to grow in the lungs versus what liked to survive in the eye? It's hard to say. Part of what our lab is actually expanding to is now we've started collecting metastatic tissue from some of these patients who had originally enrolled in our living library. And so then one of the next phases of our research is actually in the lab. Can we compare your individual eye tumor to your individual liver tumor, so that it's not necessarily just a broad primary tumors look different than metastatic tumors comparison, but what are the precise changes that happened in an individual case-by-case basis that make your metastasis different? And does that change how your metastasis might respond to a drug versus how your primary tumor might have responded to a drug? So I think this is a really good area that we're going to be learning a lot about in the coming years.
Okay, I love that. I think that's just kind of where my confusion has been, is it's like, okay, but if we know it works for the eye tumor, but then the liver tumor behaves differently, how do we know it will work for the liver tumor? So I love that you guys are figuring that out. You're in the process, like it's on the table of, like, we know this is an issue. We have to figure this out.
Yes, I think that's such an important question. And, then the next question, right, is going to be, okay, if you have an eye tumor and you're high risk for metastasis, and we know that your eye tumor looks like this, can we predict what your metastasis or your micrometastasis might be and how that might be different, so that we can give the right drug? Because we may not have a biopsy sample of a metastasis to test at the time we'd really want to be treating.
Yeah. Well, and so then another question to kind of go into that like idea is, okay, well, if the goal is to say we've got a primary tumor, we know in the eye it was high risk, so therefore you have a high risk. We have all of this data that says you have whatever percentage it is that you could metastasize in the next 2 to 5 years or whatever time frame. How do we then, I mean, how do we then make the justification for the treatment when there's nothing to see, right? Because, and I guess that comes back to the circulating tumor cells and the blood biopsies and those kinds of things to be able to prove, no, we see it here, we need to treat before it shows up in the liver, because obviously, you know, and we know, like once it gets to the liver, it becomes much harder to treat historically. Maybe not for everyone, but for the majority of people. It's not hopeless by any means, but it just, if we could get ahead of it, that would be so powerful for sure.
Yes, I think you're exactly correct. I mean, it's going to be all of these things together really inputting into the future. What's your risk? Monitor your blood. The moment we see a spike in that molecular marker in the blood, then give you the right drug that's individualized for your tumor.
No, that's the ideal, right?
That's
Nip
the
it in the
ideal.
bud.
It's a lot of work to get there, as we all know, but this is why we need to work together. And this is why we're so thankful for our patients who contribute to our research. And we're so thankful for each other because we all have different expertise and different little pieces of the puzzle that we can put together.
No, for sure. Well, and like you said, it's continuing to grow and escalate. And like there's more data each time. And just more people who are looking for kind of the same thing and just try and I guess it's kind of like if you're doing a puzzle, like if you have more people looking for the puzzle piece, like you're more likely to find it eventually or you'll find a different one that fits better and fits in a way that you didn't expect, and it somehow still is helpful. Because I know that can also be a problem when you do a puzzle, is sometimes you need another piece to be able to fit the piece you're looking for.
Yeah, I think you're so right on that. And, I think the scientific community has realized this too, and they've put a lot more emphasis recently on things like team science awards, because they know that science, the best science, probably doesn't happen in a vacuum. It really happens when people with different expertise get together.
No, for sure. Well, Dr. Davlin, this has been awesome. Was there anything else that you felt like you wanted to cover?
There were just a couple of exciting things potentially down the pipeline for uveal melanoma treatment that I can touch on really briefly.
Yeah, for sure.
ARVO, because it's more of a basic science conference, this stuff is really, really early. So this is kind of a far cry from what our other doctors were talking about earlier today that's really ready for prime time for patients. But we had some really interesting findings. We had a group from AACR out of Germany, and they talked about the impact of hyperglycemia, which I thought was really cool. They found in uveal melanoma cells in the lab, if there was too much sugar exposure, essentially, this induced abnormalities in chromosome 3, which we know kind of almost goes hand in hand with that BAP1 loss, that high risk class 2 status. So potentially blood sugar control could play an important role. And again, this is very, very early data, but we know already, especially for diabetic patients, having blood sugar under really good control has so many other important health benefits anyway. So it potentially just adds another reason to really work on blood sugar control, because it potentially could create a less aggressive uveal melanoma pattern. And along that same exact line, from MD Anderson, they looked at the insulin-like growth factor pathway and found that it might be overactivated in uveal melanoma. So blocking that pathway actually killed uveal melanoma cells in the lab. So, this is just, I thought, really potentially accessible information. That's such an easy lifestyle type of intervention that we can kind of think about and conceptualize and apply to our everyday lives. And, it's hard to know for sure how the data are going to pan out, but it doesn't hurt because it's so good for other things in our life anyway.
Oh, yeah, it's absolutely so beneficial. And I'm probably kind of speculating here, but have you ever heard of studies that were done around cancer patients that they leveled their blood sugar, and they found that cancer patients, I believe it was urine, was sweet smelling, and that it was very similar to a presenting diabetic patient, and that was kind of where that blood sugar control came into play. I mean, I'm sitting here wearing a CGM, so also controlling my blood sugar. And like you said, like it's something that diet management, and it's not even just food, right? Like eating to your blood sugar doesn't mean you're never eating sugar. Like I ate a cupcake the other day, like full disclosure. But it just means that you are watching for those patterns and you're trying to keep things regular, and you're learning to eat in accordance with what keeps your blood sugar more stable, as opposed to letting it do this all the time. Because, you know, and like you said, that's only going to benefit you as a person, because when you experience those kinds of spikes, you're tired all the time, your energy levels are low. Like there's so many different benefits aside from the potential that it might help. Like, because like you said, the data is still early, but if it might help, then like, what's it going to hurt?
Exactly.
Well, I think that's really cool. So the IGF blocking, do they have any anticipated ideas for like what can block the IGF pathways?
Yes, they have an inhibitor that's got a very molecular name yet, so it's not yet in a fun drug name status. But yeah, they did have something that they were actually able to treat cells in the lab that seemed to cause uveal melanoma cell death, and they actually correlated this too with data from the Cancer Genome Atlas, which you know has been a really nice registry that took 80 uveal melanomas and analyzed them on an incredibly detailed molecular level. And they also, you know, found again that pathway was just overactive in these aggressive cancers.
So that was called the Cancer Genome what?
The Cancer Genome Atlas. So it's TCGA. If you ever Google that, there's a really, really high level detailed paper in Cancer Cell, but it's really fantastic data.
No, I love that. I just wrote that down because I'm like, that sounds like something I want to read. I guess what was the other part that did anything happen at ARVO, or have you heard anything around Aura Biosciences and their upcoming treatment of uveal melanoma to the eye with their specific treatment?
Yeah, we're all kind of gearing up for the next phase of this trial. So at Mayo, we're going to be participating in the next phase of this trial as well. So we're just kind of waiting on the start date and the finalization of all of the paperwork here. But as you may know, historically, this has been potentially a promising treatment for really small uveal melanomas, especially things in the macula where your center of vision is, that we would love to avoid irradiating because we'd love to avoid the vision loss that comes with that. So this involves injecting nanoparticles into the eye that can tag themselves to uveal melanoma cells, and then applying a photodynamic therapy laser that activates those nanoparticles to kill the uveal melanoma. So they've kind of changed up the delivery method so that we're going to be hopefully targeting this a little more directly exactly to the tumor cells. And I'm really excited for hopefully in the next couple of months, we can start enrolling for this.
Oh, that's super exciting. So do you happen to know the trial number that people could look up if they want to learn more about that one?
It's a good question. I don't
Okay.
have that off the top of my head.
I'll see if I can find it out. I know it's the Aura Biosciences, but I just don't want to mess up the number. I think it's AU and then something. That's all we got from my brain this morning. No, Dr. Dalman, this has been so cool. And I share your excitement. I think this is really exciting stuff to just see where things are headed and to kind of see that landscape playing out and just building itself and building on itself, like based on previous research and just anticipating where it could go next.
Yeah, I can't wait to see how this plays out. I'm in this field for a long time. So, I expect by the time I'm getting ready to retire in several decades from now, that we're going to have cures for this disease. I really think that we can do it.
Well, I think that's such a hopeful note for us to end on. Is there anything else that you want to add as we end?
I don't think anything else. I just want to thank you. And again, thank our patients. We're here for you. Anything that we can do for you, any information we can provide, that's our passion. And we want to see all of our patients survive, beat this. And so we're going to work together to get that done.
Well, thank you so much for all of your hard work, and to all of our doctors who came on previously on a Saturday morning of all days. So thank you for being here. I know summertime, it can be kind of hit and miss, but we're really appreciative of you guys helping bring this information and just break it down for us as patients so that we can understand it. For those of you continuing to listen, or those of you catching the recording, please don't hesitate to reach out to us if you have specific questions for any of these doctors who spoke the last couple of hours. If you want us to pass those questions on, we can pass them on and just try and get a little bit better of an answer if you're not able to get it from the presentations. Last note is just a reminder that it is recorded. So if for whatever reason you didn't catch all of this, or you caught on for the last couple of minutes, please know that it will be up by the end of Monday for our YouTube channel, on the AACR Insight YouTube channel. And then you can also find it on the I Believe podcast in the next few weeks, once we get the audio edited. So on that note, I think we will go ahead and excuse everybody and head on out to the rest of our weekend. All right. Thank you so much for joining us, guys.